Hello everyone, I am Rahmat Hidayat. Currently I am a master's student under Dr. Al-Khadi's supervision. Today I'm going to present to you my poster entitled 16-RS hydroxyeicosahexatetraenoic acid 16 heats modulate human CYP1B1 through transcriptional and allosteric mechanisms. Well, to begin with, many studies have depicted the contribution of CYP1B1 and its associated cardiotoxic methane heats in the pathogenesis of cardiac hypertrophy. This fact drove our attention to 16 heat. It is one of the subterminal heats generated after CYP hydroxylases metabolize arachidonic acid on the chiral carbonatum number 16, producing two radioenantiomers, 16R and S heat. We hypothesize that 16RS heat will alter CYP1B1 gene expression and activity in concentration and time-dependent manner, and therefore aimed this study to determine the effect of 16RS heat on CYP1B1 gene expression and activity in vitro. Initially, we incubated 16RS heat in a plate containing RL14 cell lines. Then, we measured the CYP1B1 mRNA expression using PCR analysis, protein expression using Western blot analysis, and CYP1B1 activity using CYP1B1 dependent erode assay in the cells, as well as using human recombinant enzyme and human liver microsomes as mediators. Also, EMRODE assay to investigate the effect of 16 heat on different CYP enzyme. From the experiment, we found that both enantiomers of 16 heat significantly upregulated the CYP1B1 mRNA expression in the RL14 cell lines, with the concentrations of 20 millimolar up to 5-fold compared to control, where the 16 enantiomer R promoted prominent induction compared to its radioenantiomer 16S heat. This induction was optimally achieved after 12 hours. For the protein expression and activity, as confirmatory as it seems, the 16RS is significantly induced CYP1B1 protein expression and activity. This is a very interesting result, since 16RS heat could contract both gene expression and activity. When mediated by recombinant enzyme and human liver microsome, 16RS heat also increased CYP1B1 activity, denoting that 16 heat both enantiomers were able to increase recombinant human CYP1B1 activity and human liver microsomes. However, surprisingly enough, unlike in CYP1B1, 16 heats were able to tremendously inhibit CYP1A2 catalytic activity facilitated via EMRODE assay. This directed our understanding that CYP1B1 may experience allosteric regulation, enabling it to increase metabolic formation rate in the presence of 16RS heat. Hence, we concluded that 16RS significantly upregulates CYP1B1 gene expression and activity in the RL cell lines and increased CYP1B1 activity in vitro using the recombinant enzyme and human liver microsome. Also, the modulatory effect of 16RS heat are distinct on different CYP enzymes. That brings us to the end of my presentation. Thank you, everyone.